Hello everyone. Today we will talk about epigenetics in stem cells and during their differentiation. So all the different cells in our body from the same DNA. So one of the key questions in developmental biology is how the same genome in each cell can produce vastly different cell types. So the identity of each cell type is tightly associated with its unique transcriptomal profiles, which are shaped by highly ordered gene expression programs. So this is the single-cell RNA-seq data from the hippocampus tissue, and you may appreciate the different type of cells. They display the differential expression pattern of genes. Different combination of genes are expressed in ependymal cell, macrophage, astrocyte, oligodendrocyte. They all shows differential gene expression profile. However, all these profiles are originated from the same genetic material. That's the actual questions. So myoblasts only from muscle cells, and keratinocyte only from skin cells, and hematopoietic cells only become blood cells. But all those cells have identical DNA sequences. So how can we explain this phenomenon? So Conrad Waddington coined the term epigenetics to mean above genetics to explain differentiation. So thus genetics, the DNA sequence, we cannot explain this phenomenon. So that's something, the variations on top of the genetic information, we call this as an epigenetic landscape. So here, the ball, as I explained in the previous lecture, is rolling down the belly and eventually reach to the different status of cells. So here, the pluripotent cell, this is kind of one of the cell cover image, journal cover image for the cellnet, the biomechanical analysis of the cell status. And here, the pluripotent cell differentiate toward very diverse directions toward the mesodome or endodome or ectodome, and in each step has a remarkable, unique status that define the current cell status and differentiation status. So this is the example, the polar example of identical twins. So basically, they have same genetic material exactly. Can you tell the differences between these two people? Also, the bottom is a sister, the identical twin. You may notice that although they have the same genetic information, some of their morphologies, the characteristics, is slightly different, right? Here, the shape of nose is really different between these sisters. And here, also, the shape of eyebrow is really different in this sister. So during their development, uh, somehow different uh, different event causes some kind of differential expression at some point of the development and make the changes. This is very dramatic example. This is a photo of identical twin with different hair color. And sometimes we can notice the mosaicism. Mosaicism is example of an individual with two different eye colors. Here, this dog have the brown eye on his left, but white eye in his right side. So this is kind of a genetic, the mosaicism, and that maybe also can be explained by the epigenetic event beyond the genetic information. So we can define the epigenetics as a kind of a transmission of information through meiosis or mitosis that is not based on DNA sequence. So mechanism for stable maintenance of gene expression status that involve physically marking the DNA or its associated protein. So mitotically, or meiotically heritable changes in gene expression that are not coded in the DNA itself. And these epigenetic changes are very relevant for development and the generation of cancer. Although there are several other minor epigenetic modifications such as RNA modification or non-coding RNA or microRNA, but there are two major functions of epigenetic code in the cell. The first thing is DNA methylation. The methyl mark added to certain DNA base repress gene activity. And the second one is histone modification. Histone modification is a combination of different molecules can attach to the tails of protein. So histone has a tail, and those tails are modified by various type of chemical modifications. And those modifications affect the activity of the DNA wrapped around them. So first of all, let's talk about the DNA methylation. So DNA methylation, we majorly mention about the cytosine methylation. So the 5/5 position of the cytosine can be methylated by DNA methyltransferase, and SAM, the S-adenosyl methionine, is act as a substrate, which is major source of the CH3 residue in the cytosine. DNA methylation frequently occurred in specific sequence of DNA, CPG, cytosine, phosphate, guanine. So this CPG term is coined to prevent the confusion of CG hydrogen pair between two different strands. So in one strand, the 5' sequence is cytosine, and 3' sequence is guanine. Then we call this the CPG, C phosphate G, and DNA methyltransferase recognized this CPG sequence and methylated both cytosine in two different strands. And once the cytosine is methylated, it alters the status of the chromatin. So once the methyl CPG methylation is happened, the methyl CPG specific binding protein make the chromatin more compressed, and then that makes the gene expression suppressive. So this DNA methylation suppress the expression of certain specific gene by alter the chromatin states. And if we overview the DNA methylation pattern from zygote toward certain specific differentiated cell, there is a tendency the DNA methylation is increased during the cell differentiation. So here, the totipotent status, mostly the DNA methylation is erased and clean. However, during the differentiation, specific region of the DNA segment are methylated, and unipotent, fully differentiated cell only make a specific region of the DNA are active, and other regions are suppressed by DNA methylation. So this is one of the example here, the pluripotent stem cell. The CG is not methylated. The CG sequence is not methylated. Especially this is the location of promoter. So the first ATG is here, and this upstream promoter region is highly demethylated. So CPG sequence does not have the methylated pattern. But in unipotent gene, in this promoter, there are lots of methylated CG, which suppress the activation of this promoter. So if we overview the overall DNA methylation program, there are very drastic methylation reprogramming during early mouse development. So after fertilization, the relative methylated CPG contents are quickly decreased. So the DNA methylation pattern is erased, and after the implementation, the DNA methylation is quickly increased, and embryonic ectodome, mesodome, and those are quite methylated. And primitive endodome and trophoblast, it does not consist of embryo, but it has a very slow increase. So there are different methylation pattern in different region of the embryo. That's one of the typical characteristics of DNA methylation in early embryo. The dynamic methylation and demethylation happen during the development, and those are mediated by different enzymes. So DNA methyltransferase 3B and 3A contribute to the de novo methylation. So the new DNA methylation pattern is established by these two enzymes. And DNA methylation pattern is actually transmitted to the daughter cell. So after cell division, all of the daughter cells have the same methylation pattern because of the action of DNMT1, which copy the DNA methylation pattern toward the newly replicated DNA strand. So this methylation pattern can be maintained after the cell division by the action of DNA methyltransferase 1. And naturally, if DNA methyltransferase 1 is not actively involved, those after the DNA, after cell division, the DNA methylation pattern is diluted, so which are diminished after cell division. That is recalled as a passive demethylation. So this maintenance of methylation and passive demethylation can be caused by the action of DNMT1. So certain cell, if the DNMT1 activity is quite diminished, reduced, then the passive demethylation is more higher than the maintenance of methylation. And there is also the mode of active demethylation. So DNA demethylates recognize the methylated site and actively erase the methylation sequence, methylation regions that we call as active demethylation. So active demethylation, there are several enzymes which are tightly involved. One of those are TET proteins, and TET protein is mediate the oxidation of 5-methylcytosine toward 5-hydroxymethylcytosine. So this oxidase, this TET oxidase, owns the 5-MC, the hydroxymethylation of 5-HMC, the hydroxymethylation formed, that is further processed by other oxidase such as TAG, AID, and deaminase, and those after some DNA repair process, those methylcytosine, 5M and hydroxymethylcytosine transform toward the original cytosine again. So this is the complete pathway for dynamic modification of cytosine. It's not necessary to memorize all these things. So simply, I want to overview the process of demethylation and methylation. So as I described, the DNA methyltransferase at the methylation on cytosine transform it toward the 5-methylcytosine 5-MC, and then TAP enzyme oxidize this 5-MC, make it toward 5-HMC and 5-FC and 5-CAC, and then further processing was performed by TAZ, thymidine DNA glycosylase, which performed the DNA repair, so make it toward the cytosine again to finish the complete demethylation process. So this methylation and demethylation is continuously modulate the dynamic aspect of DNA methylation pattern during the cell differentiation. So this is the DNA methylation dynamics in the pre-implantation embryo, and the primordial germ cell, the oocyte or sperm generating cells, and those cells also have the specific pattern of the DNA methylation and demethylation. So here is a case of the embryo. The paternally methylated pattern is quickly erased after the fertilization. That is mostly mediated by the TES3 oxidase, the action of TES3, and this TES3, the action of TES3, because of the action of TES3, the 5-HMC level is quickly increased during the demethylation. And then after formation of blastocyst, the de novo methylation pattern is the newly established methylation pattern is increased in the embryo. In the case of the primordial germ cell, the demethylation the methylation is lost. The actual mechanism is not very well identified yet, but in the middle of the embryogenesis, the oxidation is mediated by TAT1 and TAT2, and the 5-MC is increased, and then they are further demethylated. So the methylation pattern of primordial germ cell is relatively maintained in very low level during whole embryo development. And DNA methylation and histone modification is actually work together to help to compartmentalize to the genome into domains of different transcription potential. So certain region of the genome form the euchromatin, which contain the high histone acetylation and low DNA methylation and high level of H3K4 methylation. And this euchromatin is relatively loosely maintained and contains many active regions of the transcription. And heterochromatin also formed in certain region of the genome, and this heterochromatin contains low histone acetylation and make the very compact structure, and those have the very dense DNA methylation and have a high level of H3K9 methylation. So the balance of the euchromatin and heterochromatin must be tightly regulated and modulated during the cell differentiation, and those are the combinatory action of DNA methylation and histone modification. So this is an example of the post-translational modification of histone tails. As I described, the histone has a tails which is located outside of the chromatin, and these tails are modified with various post-translational modification. For example, the phosphorylation happen in arginine rigid or ricin rigid or many different rigid of the histone H3 tail, and histone H4 tails are also modified. And 2A tail and 2B tails are modified, and among those modifications, which contains a phosphorylation, acetylation, methylation, and ubiquitination, and various type of these post-translation modification modify the characteristics of the histone tail, and also recruit many the histone modification specific binding proteins which alter the properties of the chromatin. So here is the example of the tail modification of histones. The acetylation, methylation, phosphorylation happen in various regions of the histone tails. And DNA methylation, in the case of the DNA methylation, the writers are DNA methyltransferase and erasers are TATs, as I described before. And also, there are writers of the histone modification, specific histone modification, and erasers of histone modification. For example, polycomb complex 2, the PLC2, is a writer complex of the K27 methylation. And also, this methylation can be erased by UTX1 or JMJD3, demethylase of the histones. And also K9 methylation or K4 methylation. Also, there are writers and erasers to enable the dynamic change of these histone tails. And we also we previously talked about the HAT, histone acetyltransferase, and HDA, the histone deacetylase, which is important to regulate the compactness of the chromatin, and which is mediated by the K27 acetylation. Again, I would like to emphasize it's not necessary to memorize the name of these things, but we need to memorize the concept of specific writers and eraser enzyme, which dynamically regulate the post-translation modification, which is very critical for the status of the chromatin and functionality of the chromatin, and the level of active transcription and inactive transcription. So this is very landmark paper, which is published in the Eric Renders group in 2006. Maybe this is an example how the various chromatin histone modification regulate the status of the chromatin, which is critical for the development of cells. So the question is how we can analyze the post-translation modification pattern of the chromatin. The powerful technique developed for this purpose is chromatin immunoprecipitation, CHIP, and accompanied by the high-throughput sequencing technology. So after the cross-link of protein to DNA, it's a quick cross-link. It's kind of the fixed protein location toward the DNA, and and then the scientists shear the DNA by sonication or enzymatic degradation. So this fragment, the complex of the DNA binding protein and DNA are fragmented, and then this sheared DNA strand can be pulled down by bead-attached antibodies by the immunoprecipitation. So applying the specific antibody for a specific modification on the protein, we can pull down the DNA fragment which is associated with these modifications, this antibody. And this DNA sequence are generated by the high-throughput sequencing library, and then we can sequence those DNA to map to the genome. And after this kind of a ChIP-seq experiment, we can enrich the specific DNA sequence which is associated with a specific type of histone modification, because in this case we use the antibody against the K4 monomethylation, and here is K4 trimethylation or K27 acetylation antibody. So we can critically pull down the DNA fragment which is associated with this specific type of modification. So these peaks represent the frequency, the probability of this modification which exists in this genome. So here, this region contains more higher level of K4 trimethylation compared to the other region of the genomes. And this peak represent this region contains high level of K27 acetylation. And we can define this histone code to the genomic sequence, and this genomic histone code represent different activity and different status of chromatin. For example, there's a bivalent promoter which carries activating marker, the K4 trimethylation, or repressive marker, K27 methylation, in simultaneously. For example, this region have the bivalent promoter which have the repressive marker K27 trimethylation, as well as active marker K4 monomethylation as well. So and there's a enhancer, which is enhancer region is mostly marked by the K27 acetylation as well as the K4 methylation. So these regions have the high level of K27 acetylation as well as K4 methylation. So we can define this region is active enhancer. And this is active promoter, which have the high level of the K4 trimethylation as well as K4 methylation, but it doesn't have the high level of K27 acetylation. So we define this region as active promoter. And there is a poised enhancer. Poised enhancer means the closed enhancer having K4 monomethylation with repressive marker K3, K27 methylation, and it doesn't have the K27 acetylation mark. So this is the potential enhancer, but it doesn't actually actively involved in the transcription. So we call this as a poised enhancer. There are multiple various descriptions. So bivalent poised promoter is marked by the K4 trimethylation as well as repressive marker K27 methylation. These poised genes, RNA polymerase II is located at the promoter in the absence of ongoing transcription. So polymerase is halted, stuck there, but do not perform the active transcription. The genes are loaded to be transcribed soon. So this is kind of ready, the region which is ready for the de novo transcription once there is some stimulation applied. And this is active promoter, the K4 trimethylation and K27 acetylation mark the active promoter, which the gene is actively transcribed. And there's a various type of definition of the histone mark. For example, this region has a repressive marker K27 trimethylation, as well as the active marker K4 methylation that we call this poised, which are balanced developmental enhancers. And the K27 trimethylation enriched region is tightly repressed region because this K27 methylation is mediated by the polycomb repressed complex, so we call this a polycomb repressed. And K9 trimethylation is heterochromatin. This is a very tightly suppressed region of the transcription. So let me go back to the original paper, the Eric Rander's paper, the bivalent chromatin structure, mark key developmental genes in embryonic stem cell, which is published in Cell in 2006. And at that time, they didn't have a high-throughput sequencing technology, so they used a chip microarray. But basically, the concept is the same. It's a cross-link and sonication to fragment the DNA, and then using specific antibody, in this case specifically the K4 methylation antibody as well as K27 methylation antibody, which is represented for the active chromatin and inactive chromatin. And then they hybridize these fragments to the microarray, and then examine the frequency of existence of these marks in the genomes. And then they found the interesting thing, when they use embryonic stem cell, this embryonic stem cell has very specific characteristics because they compare these embryonic stem cell histone markers, active and inactive. So this yellow region mark the active region, inactive region, and this green region mark the inactive region. And this red thing, red marked locus, implicating the both inactive marker and active marker coexist together. So we called it as a bivalent domain. Both active and inactive markers are coexisted in this bivalent domain. And this bivalent domain is remarkably highly existing in embryonic stem cell, not in other cells, such as muscle cells, or neuron cells, lung cells, and skin cells. They only generally mark the inactive regions with active regions and inactive regions with different 27 methylation and K4 methylation. But these marks do not coexist in this differentiated cell. However, in embryonic stem cell, undifferentiated cell, this active marker and inactive marker both exist. This is another example. The ES cell, there is the bivalent domain, frequent bivalent domain for the important developmental regulators. However, differentiated cell, the C212 cell, Neuro2A cell, MLK, MEF cell, they have certain reason as an inactive marker and certain reason as an active marker, inactive marker, but they mostly do not coexist together. So this is the summarization of active and inactive and bivalent domain in different cells. So ES cell remarkably have a very high frequency of bivalent domain, and there's active domain and inactive domain. However, the differentiated cells have very little amount of bivalent domain compared to the ES cell, and they mostly are marked either active domain or inactive domain. This black represents there is no pull down fragment. That means this region is unclear whether they have a methylation, these methyl markers. So don't care about this black mark. So the conclusion is ES cell contains many bivalent domains, and the second, these domains are highly enriched at developmentally important genes that establish cell identity. Transcription factors. Interesting thing is many of these bivalent domain, the blue color represent the transcription factor, and most of these bivalent domains are the genes related to transcription factor. That means the transcription factor, which is generally very important for the cell identity. So cell identity determining the transcription factor mostly have bivalent domain in embryonic stem cell. And interestingly, this is an expression pattern, the mRNA expression pattern, which is assessed by the quantitative PCL. And here, the blue one is embryonic stem cell, and the violet one is the result from the neural precursor cell. The neural precursor cell express NKX2.2 or SOX21 abundantly, and they have active markers. This is the case of the chip experiment in neural precursor cell. These active genes, the violet active genes, have the H3K4 methylation. So it's mostly active region of the active histone marker containing regions. However, ES cell, although they don't express these developmental markers, only they express pluripotent potency markers, but these developmental genes are not expressed, but they have both active marker HC3K4 methylation as well as 27 methylation, active inactive marker coexistence. This coexistence is not abundant in the case of the neural precursor cell. So they have active markers in actively transcribed genes. They have inactive marker in inactive the genes which doesn't have high expression. Although there are some kind of bivalent locus in neural precursor cell, but mostly they have active locus or inactive locus. So we can, this report suggests that ESL have maintain abundant bivalent chromatin, which keeps the genes poised, balanced, and ready to be activated. They are not activated yet, but they are ready to be activated. So these lineage committed genes, cells, the ES cell, those are actually polymerase, they're posed and adhered here, and they have both active 27 methylation and active K4 methylation and inactive K27 methylation in their chromatin. And when they differentiated, certain regions are completely off, certain regions are completely on, and certain regions are poised. These existence of bivalent domain silence developmental genes while keeping them poised for the activation in ESL. And by this way, this region can be activated very quickly when the developmental cue is applied on these stem cells, and when they differentiate it, they can actively transcribe this region very quickly according to the developmental direction. So here is the description of general epigenetic changes during cell differentiation from embryonic stem cell to neural stem cell. So during differentiation, the pluripotency genes are getting repressed by the epigenetic modifications. So pluripotency genes, their promoter is highly active in embryonic stem cell. However, during differentiation, those promoters are keep modified by repressive marker. So the promoters are getting repressed and inactivated. However, the lineage-specific genes, the developmental genes such as neural genes and endodermal mesodermal gene have differential regulation. They experience the differential regulation. So ESL, as we discussed in the previous paper, those developmental genes are poised. So they both have active markers such as ACC-K4 as well as A3K27 methylation. However, those are ready to go, but they are not, the transcription is not initiated. And during differentiation, the neural genes are selectively turned on. So repressive marker is gone and only active marker is maintained. And those promoters are active and actively initiating the transcription of the neural specific genes. And in the case of endodermal or mesodermal genes, those were previously poised in the status of embryonic stem cell, and the active marker is gone and repressive marker is further increased, and those chromatin change to the heterochromatin, which were their transcription is highly repressed. So by this way, only selected genes are activated during the cell differentiation, which make the cell specific to certain lineage. As you know, the genetic information inside of the encoded in the DNA sequence is tightly packed into the eukaryotic cell. So this is a general description of eukaryotic chromosome organization. And as you see here, the DNA double helix are coiled very tightly and wrapped around the nucleosome, the histone proteins. And these histoproteins are also the fold up to produce 30 nanometer fiber, and these fibers forms loop structures, and these loop structures are further compressed and folded to produce 250 nanometer wide fiber, and these fibers are further coiled and tightly packed into the chromatid of a chromosome. Let's go into more detail about the chromosome and chromatin structure inside of the nucleus. So this is the electron microscope image of eukaryotic cell nucleus. You may notice that there are bright region and a dark region. The bright region represent the euchromatin, which the transcription is active, and these dark region consist of the more condensed chromatin, which is heterochromatin. So transcription is generally very repressed in these heterochromatin dark regions. And you may also appreciate that this heterochromatin region is widely spread in the nearby the nuclear membrane, as well as the nearby the nucleoli structure. And in the nuclear membrane, the important protein, ramin A and ramin B, is enriched. So we called it this nuclear membrane associated chromatin as a laminar associate domain, LAD, LAD. And this ramin, nuclear membrane protein, is very useful if we use the DNA adenine methyltransferase identification. DAM-ID is very useful to reveal the LAD in the DNA region. So let me explain briefly about the adenine methyltransferase identification. Because adenine methylation is very rare in eukaryotic cells, so it's almost never found inside of the DNA. So using these characteristics, they use the DAM enzyme. DAM enzyme is adenine methyltransferase. So if we fuse certain proteins such as lamin, nuclear membrane lamin protein, together with the DAM enzyme, then this DAM enzyme transfer the methylation on the adenine nucleotide around DNA sequence. So by this way, we can easily identify the adenine methylated region as the location closely the location which was closely located to the certain protein such as lamin. So with this way, if we fuse lamin, the nuclear membrane protein, with the DAM, the adenine nucleotide, which is close to the lamin, is methylated. And if we use this adenine methylation specific binding protein, DPN1, fused with GFP, we can easily identify the chromatin region, the DNA region, which is associated with lamin. So with this way, the ramin B is represented in the red region, red color here, and this green fluorescence region is actually the adenine methylation tracer fused with GFP. So these regions represent the DNA fragment, DNA segment which are associated with nuclear membrane. So this green region is represent for the laminar associate domain, LAD. And with this way, this DAM-ID DAM-ID, we can sequence this adenine methylated region. So this is the adenine methylated region. The green region is LAD, laminar associate domain, and the blue region is inter-LAD regions, which is not associated with lamin. So here, if you look at the chromatin structure, some of the chromatin is away from the nuclear membrane, and some of the chromatin is close to the nuclear membrane, and this DNase actually goes in and out in laminar associate domain here. So this is another diagram which depict the structure of the chromosome nearby the nucleus. And here, the ramin LAD region, the ramin associate region, is densely packed as a heterochromatin, and you may appreciate that there is a lots of DNA methylation as well as the repressive marker represented in the red circle here. And this compact nucleosome exists around the nuclear membrane, and this loop deep inside directed inside of the nucleus is euchromatin, which the transcription is active, and the chromatin is very loosened and not tightly compacted. So transcription and polymerase and transcription factor can easily accessible with this active region of euchromatin. So this loop structure and Ramin associate domain is the very general characteristics how the DNA is located and assembled inside of the nucleus. Again, this diagram shows the general architecture of the chromosome inside the nucleus. So this different color, violet color and blue color and green color, pink color all represent the different chromosome. So chromosome number 1, 2, 3, 4, 5, 6 located in different region inside of the nucleus. So they are well arranged with certain order, and the nuclear lamina hold a certain DNA region as a heterochromatin, and the inside loop is more active region. And NAD is a nucleoli associated region, which is also consists of many heterochromatin region. As we observed in the electron microscope image in the previous slide. How this architecture can be formed inside of formed with in structure. So there are self-interacting domain. Those DNA as the fragments are interact each other, and this interaction is helped by mediated by the architectural proteins. So these architectural protein hold certain region of the DNA segment together, and by this way, this DNA forms some kind of loop structure, and this loop structure can interact frequently each other. And we can also define the chromosome territory. So chromosome, certain specific chromosome is located mostly in the same location. So for example, this chromatin 13 located here, chromosome 10 located here, chromosome 9 located here. And the chromosome, the DNA segment located in the same chromosome are more likely to interact together. However, certain segments interact also with other chromosomal DNA segment. So this is called as interchromosomal interactions. In general, we called open chromatin, the more actively transcribing chromatin, as an A compartment, and more repressed chromatin, more condensed chromatin, closed chromatin as a B compartment, and B compartment generally located nearby the LAD, and A compartment is located in the inter LAD, which is far away from the nuclear membrane. And DNA looping is mediated by the multiple protein interaction, which bind to the DNA as well as they interact with protein-protein interaction as well. So enhancer cis element can be interact with mediator or other proteins, and those interact with the transcription factor as well. And the promoter is also interact with the multiple transcription factor or transcription cofactors. And the CTCF and cohesion is important structural protein. Make the specialized loop architecture. We will talk about it later probably. And also, I would like to introduce the concept of a topologically associated domain, TAD. The topologically associated domain is the region which interact very frequently, tightly, and generally those TAD region is defined by, generated by the architectural proteins. So these TAD make kind of their own loop, and they interact more frequently inside of the nucleus. So if we use, if we think about the DNA interaction inside of the TAD, these genome segments interact more frequently each other, and that's because of the structural arrangement of DNA sequence. How can you identify this frequently interacting DNA sequence? So-called topologically associated domain, TAD. There are numerous newly designed technological sequencing technologies for the DAD identification, the chromosome conformation technologies. So basically, those techniques cross-link the chromatin, and after cross-linking, with multiple way they digest the cross-link the chromatin as a small fragment, then ligate them together and identify the circularly ligated region as a interacting sequence, interacting regions. So these blue region and red regions are closely located, and after ligation, these closely located segments are combined and sequenced together as a linear sequence. So then we can say this blue DNA sequence interact tightly with this red sequence or located very closely. So by this way, we can identify the DNA interaction with chromosome conformation technologies. And there are multiple variations such as 3C, 4C, 5C, high C, chip loop, chip A PET. Maybe we can talk about one of these popular technique, that is high C. So high C is after the cross-linked. As I said, the closely located regions are sequenced together, and these sequence are aligned with genome, and then those interacting regions are mapped together. So this red circle is if we find the interaction between this green segment and this pink segment, this is the probability of this cross interaction is mapped as a red square here. And if this yellow region and the blue regions are sequenced together, and this increase the probability of this square, red square, in this two-dimensional map. So here, if we see this red dot in this location, that means the chromosome in chromosome 20, this sequence and sequence here is found together as a single lead in linear genome sequencing. So that is this region, and probably region around here, they interact together. And if this dot is found, that means the region here and region here is interact together. That means somewhere around here is interact tightly. These DNA, two regions of DNA interact together. So this is the way how we read the high C map. So here is a description of the red square, the meaning of red square in structure. So if we find squares around here, this means the subcompartment interact together. So the region around inside of this subcompartment are located like this. So they interact frequently together because they are structurally located closely, right? So these are the meaning of these squares here, right? The subcompartment. So this is kind of the chromosomes are located together in a very similar area. And let's amplify this region. And if we amplify this, this is more microstructure, the ordinary domain. So the ordinary domains are inside of this loop. So they are located in the almost same loop. And if we amplify this more, the very dense, intense dots on the on the point of a square, that is we call it as a loop, and loop is kind of tightly connected by the cohesion or CTCF, so the architectural protein, most architectural protein in general. So these dots in the high-C maps represent the loop formation of this DNA region and this DNA region around here. So there is a kind of very long-range interaction between these two regions through this loop formation. So these type of chromatin architecture are dramatically changing during the cell differentiation, especially from the pluripotent stem cell. And the reprogramming also alter the dramatic change, alter the structure of the chromosome very dramatically. So here is one example. So in pluripotent cell, the pluripotent genes are actively transcribed, such as OCT4, SOX2, nanogs are actively transcribed. So the promoter of the OCT4 is occupied by transcription factor and also polymerase and enhancer binding proteins, mediator. So those transcription complex are formed actively in the pluripotent cell. However, during the differentiation, this loop structure of the chromatin is changing. So these CTCF and cohesion proteins are moving, sliding toward other direction, other locations, and make the loop structure altered. And then this region, the octuple promoter region, and enhancer region cannot interact together right here. So this blue enhancer and black promoters can interact very easily because they located closely with this conformation of the chromatin. But after the differentiation, these loop structures are changing, so the enhancer and promoter regions departed far away, and also this enhancer or promoter regions are repressed by diverse histone markers such as 27 methylation or H9 trimethylation. So the heterochromatin forms nearby the enhancer and promoter, so these pluripotent genes cannot be activated. Previously, during the process of the reprogramming by expression of OSKM in the differentiated cells, this structure of the chromatin is remodeled, and the enhancer and promoter can be approached together after the reprogramming to activate the transcription of pluripotent genes. So this one remarkable paper is studying the local genome topology through the somatic cell reprogramming, during the somatic cell reprogramming. So they use the embryonic stem cell and differentiate those embryonic stem cells toward the neural precursor cell, primary neural precursor cell. And then they reprogram this neural precursor cell again by the expression of OKM reprogram factors. So from ESL, the chromosome architectures are remodeled toward the MPC status, and then again the consequent reprogramming make the MPC as toward the induced pluripotent stem cell. So during this process, they examine the chromosome structure changing by the high-C techniques, and this high-C maps shows various changings of chromosome structures. And with amplifying the high-C result, so this is the whole genome result, and if we amplify the one chromosome region, the chromosome 3 is here, and also amplified more, this 10 megabase pair surrounding SOX2 locus, which is important pluripotency marker, and also 1 megabyte surrounding, so it's very high resolution. The one being represent the 4 kilobase pair, which is remarkably very, very fine for the high-C maps. So if we amplify the 1 megabyte pair surrounding the SOX2 region, the ESL, MPC, and iPSC, so there is very dramatic changes, and you may notice that the profile of the MPC is different with ESL, and ESC profile is quite similar with iPSC. So that means during the reprogramming, differentiation, and reprogramming, the chromosome architectures are dramatically remodeled, and reprogramming process from MPC to iPSC restore the original conformation of chromosome, similar with the ESC. In the case of nanogu, you may notice also similar thing. The ESL MPC shows very dramatic profiles. However, after reprogramming, the shape, the formate, the structure, and the heat map of the high C data is quite similar with iPSC. So this is the amplified one. So ESL, the MPC, the ESL, if we differentiate ESL toward the MPC, the neural precursor shows very dramatic, very different conformation revealed by the high C heat maps. But after reprogramming, this dense cluster in the center is restored after reprogramming, and many type of the DNA interaction, the chromatin interactions are restored after the reprogramming. So this is kind of a cell type specific architecture of the chromatin is revealed by the high C data. And if you have very keen eye, you may also notice that not all of the MPC profiles are restored toward the ESC profile after the reprogramming. So here in iPSC, although this region is fully restored, very similar with ESL, and many other regions are actually quite restored. However, if you see this square, here the ESC is highly interact together, but MPC, these regions are not interact very much. And after reprogramming in iPSC, this region is partially interact frequently, but not as much as the thing happened in ESC. It's very dense frequency happened here, but it's not fully restored. You may notice also other regions, such as this region. It's quite blank, but those are quite interact together here, but these interactions are not fully restored, so there is still less interacting together. That means the pluripotent genes can exhibit persistent MPC-like folding patterns in iPSC. So MPC-specific folding patterns still, some of them are still maintained in iPSC after the reprogramming. So that represent the incomplete recovery of 3D genome architecture of ESL after reprogramming. So we can say this type of reprogramming is not perfect. So some of the MPC-like epigenetic pattern or folding pattern of the chromosome folding pattern is still persist after the reprogramming. Let me introduce briefly. I didn't mention the naive status of the ESL in the previous lecture, but there are two different status of embryonic stem cell. The first one is naive state, and the second one is primed state. As you recall from the name, the primed status is more differentiated ESL status. So human ESLs are primed status in the ordinary culture. They have one X chromosomes inactivated. And there are some kind of media condition, which make the stem cell more naive, undifferentiated cells. So here you can see the shape of colonies are relatively flat in prime status, and shape of colonies are more dome-like round structures in naive states. And in naive states, it's more primitive states. It's more early embryo. So primed states is mimicry of the cylinder cup-like status. Is relatively later status compared to the blastocyst-like structures. So this is blastocyst-like status. Naive status represent more earlier stage of the development. And this naive status can be achieved by simply addition of inhibitors chemical inhibitors on the media. So which is where simply we can we need to change the media composition toward the naive status. We can easily transform this primed ES cell toward the naive ES cell. So they apply these two eye conditions, two inhibitor added conditions toward the iPSC and perform high-C experiment again. An interesting thing is this region present in ESC, and this is disappeared after MPC differentiation, and those are partially reprogrammed, so not completely restored, but partially restored. But if we add 2i media to make the iPSC toward more naive status, these regions are restored. Here, this more fine structure. So this ESL, this is present. MPC, this is disappeared, but this interaction is only partially restored after the reprogramming from MPC. However, if we add the 2i media to make the iPSC more primitive, more naive status, this ESL-like the chromatin folding is restored. This means structurally ESC, this region make a close interaction by these loop structures. However, MPC, this region is folded another way. So this black region does not interact together. However, after the reprogramming, is partially reprogrammed, so some of the MPC-like structures are still maintained. However, this MPC-like folding pattern is disappeared when we add the 2i inhibitors into the media to make the iPSC more naive status. So reprogramming is so complete reprogramming is more facilitated, more promoted by the condition of the cells more primitive after the reprogramming. So an interesting thing is the pluripotency factors like the reprogramming factor, OSKM, facilitate the interactions of the newly formed chromatin interactions and change the chromatin organization to facilitate the reprogramming. So here is the description of differentiated cell, and differentiated cell, the interaction of DNA segments. DNA segments are not very tight. So enhancer and promoter, they are located closely, but they are not fully tightly interact. However, after reprogramming, if we overexpress the pluripotent factors or the endogenous pluripotent factors are turned on, they recruit the chromatin architectural protein and also fortify the interaction between this long-range contact, especially for the promoter and enhancer, and the pluripotent other type of pluripotent genes or stem cell maintaining genes are turned on with this way. So pluripotent factor also contribute the dramatic chromosome reorganization, which make the cell more naive and primitive, eventually achieve the pluripotency. So let's go back to the Waddington manuscript representation. We already discussed a little bit, but this is the path of normal development. So it's just go downward through the belly, and cells are differentiated. Reprogramming toward the pluripotency is by expression of the reprogram factor. We can roll upward these blue balls toward on top and make the cell pluripotent. And we can also consider about the direct reprogramming. So change the one cell status toward another lineage cell by alter the epigenetic landscape of the certain cells. So we can change the fate of cell from one fully differentiated status toward another fully differentiated status. We call this as also transdifferentiation. So differentiate one status toward another state. let's talk about this direct reprogramming method using the epigenetic factor or transcription factors. So the concept of direct reprogramming, as I said, it just changed the one cell status toward another cell status. And we can also think about the indirect reprogramming, so make one cell status toward more undifferentiated status, such as a progenitor status, and differentiate this progenitor toward another cell type which can be naturally generated from this intermediate progenitors. So this is a strategy. So if we add certain type of transcription factor into the fibroblast, those transcription factor induce the transdifferentiation or direct reprogramming toward neurons, which express the neuron-specific markers. We call this induced neuron, IN, and these neurons can be specified in various subtype of the neurons. And if we add another combination of transcription factor, which promoting the reprogramming of fibroblasts toward neural stem cells, we call this induced neural stem cell, which express the neural stem cell markers such as NES and SOX2. Then this is neural stem cell, so we can generate diverse type of neural lineage cells such as neuron and astrocyte and oligodendrocytes. So with this way, we can push up this ball toward the progenitor status and push down toward another lineage cells such as astrocyte. We can generate the fibroblast toward this INSC, neural stem cell, and generate diverse type of cell in the neural lineage cells with this indirect reprogramming. And direct conversion, direct reprogramming, facilitate the differentiation, the conversion of cells from fibroblasts toward various lineages, such as neural stem cells with various factors, dopaminergic neurons, and GABAergic interneurons, neuronal cells, cardiomyocyte, hepatic stem cells, hepatocyte, hematopoietic stem cell, or renal tubular epithelial cell, and ordinarily, we can also reprogram fibroblasts toward the induced pluripotent stem cell by OSKM factors. So many groups are studying the direct conversion using the various transcription factor or epigenetic modulators to specified cell types. And this is another description of direct reprogramming. So we can change the fibroblast toward the muscle cell and megablast, which producing the erythroid megakaryocytic cells, and macrophage, endocrine cells, fat cells. Many cells can be converted from diverse cells, and not only just fibroblasts, almost precursor cells are converted to the fat cells, or exocrine cells converted to the islet cell. T cells can be converted toward NKT cells. It will be very helpful to understand the direct reprogramming explained by Marius Rogni, who developed initially the direct reprogramming system for the neuronal differentiation that are induced to neurons.
My lab is interested in the fundamental question of what a cell identity is. You can imagine you have a ton of different cell types in your body. You have skin cells, you have brain cells, you have heart cells, you have liver cells, and so forth. And all these cells have the exact same genome, yet they look very, very different. We are really curious to understand what are the mechanisms that determine these different identities. My lab has shown that it's possible to convert skin cells directly into neurons. And the surprising thing was that those neurons really behave just like the neurons in our brain. So this is really exciting because we can now generate human neurons in the culture dish, and we can use these cells to repair brain damage And also we can try to use these cells to, when we get these cells from patients, to try to capture disease processes in the culture dish.
To find the transcription factors which transform the fibroblasts toward the directly neurons, the Marius used very similar techniques which the Yamanaka first used to develop the induced pluripotent stem cell. So they used a mouse model which have the EGFP expression under the neuron-specific promoter, so tau promoter only active in the neurons, not in other type of cells. So those neurons only exist in the central nervous system, the brain, and spinal cord. So they remove the neural tissue from this E14.5 embryo and only isolate the fibroblast from the skin. So this skin derived fibroblast doesn't express a GFP. Because they express only genes which is suitable for the fibroblast identity, and they express the candidate transcription factors, many candidate transcription factors, and incubate culture these cells in vitro for 12 days, and they found some of the cells expressing GFP. That means the neuron-specific promoters are turned on. So they isolate these colonies and find the transcription factors, make the more efficiently generate the green reporter positive cells. So by this way, the non-infected mouse embryonic fibroblast does not express GFP at all, and the TUJ1, the neuron marker, is rarely expressed. However, 32 days after 19 factors they selected many transcription factors altogether, so 19 select factor expressed. They found that GFP reporters are turned on, and also the neuronal marker TUJ1 is expressed after these 19 factor expressions. So they minimized the factors one by one and found the 5 factors are essential. So 13 days after 5 factors, BRN2 and MYT1L and GIC1 and olig2 and ASCL1 transformed fibroblasts toward the neurons expressing GFP protein as well as the neuron-specific markers within 13 days. And they found that electrophysiologically, these IN, the induced neurons, have the activity, the membrane potential, and the electrical activity, which can be stimulated by the activity can be stimulated by the electrical pulse. So these properties showing that these transformed IN actually have the functional properties of neurons. So they minimize the factors further. From 5 factors, they minimize the factors toward the BRN2, ACL1, and MYT1L is sufficient to generate the IN efficiently. Although other factors can make the IN, but this bump factor mediated IN shows the most highest the efficiencies of the transformation. And these bump factor induced neurons shows very high functionality revealed by the electrophysiological recording. So why the direct reprogramming is beneficial? There are many differences between these two methods. Making induced pluripotent stem cell from somatic cells and differentiate those pluripotent stem cell again toward the neurons or other cell types, disease-relevant cell type. This pathway, the differentiation from iPSC, takes time. It's quite a long time. Like a fibroblast, we need to more than 3 weeks to reprogram the cells, and also we need to amplify the identified induced pluripotent stem cell, and we also need to differentiate certain types from the iPSC. It takes quite a long time. However, the good thing is we can increase the cell number by proliferating induced pluripotent stem cell, so we can have unlimited cell numbers for the experiment or treatment or cell therapy. Direct conversion is very beneficial because it takes very short time. So by expressing the transcription factor or epigenetic modifiers, we can easily transformed fibroblast towards certain cell type which is relevant for the experiment or therapy, but it has a limited cell numbers because if the fibroblast is not sufficient to generate numerous cells, those are limited by the number of fibroblasts, because fibroblasts cannot proliferate infinitely. So this strategy, somatic cells are directly converted to neurons, and we can generate the fibroblast-derived neurons. So it skipped the development. Is actually age equivalent. That means if we use the old fibroblast, it has all the signature. The direct neurons have all the signature of aging. So from aged fibroblast, we can generate aged neurons, aged induced neurons. However, if we use the induced pluripotent stem cell, by generating the induced pluripotent stem cell from somatic cells, those aged signature was all gone, all rejuvenated. So these neurons are still very young. It has the characteristics similar with the young neurons instead of the aged neurons. Although we use the aged somatic cells, it has actually good things and bad things, because we can generate the young neurons, which is good for the transplantation therapy or cell therapy. However, with aged neurons, we can study their signature and phenotypes of aged neurons, such as if we want to study the neurodegenerative disorders, such as Alzheimer's disease, it's more beneficial to generate the induced neurons from the aged fibroblast to study the degeneration of those neurons instead of the very young neurons, because very young neurons are very healthy and resistant for the degeneration. And this is the description of diversity and mosaicism. From the iPSC reprogramming, we can escape from the mosaicism, because although the primary fibroblasts have some mutations or different characteristics, if we clone the iPSC by picking up the appropriate colony, we can generate kind of a clonal identical iPSC line. So those neurons may have more homogeneous characteristics. If we convert the heterogeneous fibroblast culture toward the direct induced neurons, the induced neuron have to still maintain the mosaic characteristics of fibroblast. So its genetic diversity can be more relevantly represented by the IN cultures, because if we want to study the somatic mutations which present in the fibroblast, then this strategy probably more relevant. If we want more consistent result, getting the more consistent results from the experiment, this iPS-mediated strategy is more relevant for those experiments. Not just for the neurons, we can generate diverse cell types, such as cardiomyocyte. And cardiomyocyte also can be generated by direct reprogramming. So here is the fibroblast, and by expressing several transcription factors such as GATA4, TBX5, MEF2C, we can convert fibroblasts toward the cardiomyocyte. And also, traditionally, if we convert the fibroblasts by the reprogram factor and make the iPSC, and using some certain type of cell culture techniques, we can differentiate this iPSC or ESC to cardiac progenitor cell and toward cardiomyocyte. And let me talk about the in vivo cellular reprogramming. So in vitro, we can generate with certain transcription factors, the fibroblasts can be converted toward cardiomyocyte-like cells in vitro in culture dish. And so minimal TF transcript factors were identified. Only 3 factors like GATA4, MEF2C, and TBX5 is sufficient to induce the cardiomyocyte-like cells from the fibroblast. Interestingly, if we inject these factors using the retrovirus, if we introduce these factors into the endogenous, the in vivo fibroblast, the induced cardiomyocyte can be generated, which have the properties of electrical coupling. they just trembling and move together, use the electrical coupling, and it can decrease the infarct size when the cardiac tissue has infarction. Those actually those tissue are damaged and transformed to the fibroblast. Those infection size can be minimized by retroviral injection of these transcription factors, and the cardiac dysfunction can be attenuated, and it shows the potential to be used as a therapeutics. Directly programming can be used for the therapeutics for the cardiac disorders. So the cardiac reprogramming largely help for the heart repair. So if certain region is injured by the infarction, this damaged region usually generate the cardiac fibroblast around, and this cardiac fibroblast can be directly reprogrammed by the introduction of these factors. And from this cardiac fibroblast, it can restore the damaged region of the heart by the in vivo reprogramming, using the virus expressing reprogramming factors. A similar strategy can be applied for the liver disease. So using when the liver is damaged harshly, this damage increase the fibrosis. So the fibroblast-like cells are integrated into the liver and make the liver more hardened. And this myofibroblast, if the adenovirus delivered toward this myoblast with the transcription factor overexpression, such as FOXA3, GATA4, HNF1A, and H4A, the in vivo myoblast can be converted with induced hepatocyte cells, hepatocyte epithelial cells, and that diminish the amount of the fibrosis inside of the liver, and therapeutically beneficial for the liver disorders. So in vitro reprogramming is also possible. So antivirus, using the antivirus, myoblast can be converted to the IHEP cells in vitro, and we can use these two-dimensional cells to investigating the mechanism of liver disorders, or screen the drug for the liver therapeutics. And also similar idea is also useful for the neurodegenerative disorders. So in the case of the Alzheimer disorders, the glial cell, usually the neuroinflammation, the inflammation in the nervous system, increase the glial cell population, and this glial cell can be converted to the neural precursor cell by the expression of the transcription factor or neuroblast, and eventually it can be differentiated toward the neurons. There is examples of in vivo reprogramming of astrocyte to the neuroblast in the adult brain, and in vivo conversion of astrocyte to neurons in injured spinal cord, because the injury also overproliferating the astrocyte in the injury site. So if we can convert these ectopic astrocyte toward the neurons, maybe it is helpful to cure the injured spinal cord. In the similar strategy is in the case of the Alzheimer disease model, the brain injury, the reactive astroglial cells can be converted into functional neurons by introducing the transcription factors in vivo by virus-mediated delivery. This is also the pancreatic exocrine cell can be reprogrammed toward the beta cells. So the insulin producing beta cells can be generated by the introduction of transcription factors. So there are many type of insulin producing cells after the introduction of this virus expressing the transcription factors. You can see the green dots and red dots in the islets. So this type of direct reprogramming can be used for the treating the various type of disease in vivo. So let me summarize the diverse approaches for the cellular programming so far we have learned. So there is a somatic cell nuclear transfer to change the clone, change the somatic cells toward the pluripotent cells. And sometimes the nuclear extract is working to change the somatic cells toward the ES-like cells or progenitor cell. And cell fusion of the embryonic stem cell and somatic cells can be used for the generation of ES-like cells. And defined factors such as Yamanaka factor, OSKM factors transform the somatic cells toward the pluripotent stem cell. And today we have learned the direct conversion. The somatic cells can be reprogrammed directly with the cell type specific transcription factor toward a certain type of differentiated cells. And in vivo reprogramming is also possible. So by the delivery of transcription factors, we can generate these designated cell types inside of the whole animals, and those are very useful for the therapeutic approaches in the future. So Waddington band can be manipulated by various ways. So now we are developing the ways to manipulate the differentiation paths and reprogramming paths by various techniques. And then we can now our objective, one of the future objective is generating the different cell type freely and very safely, which is applicable for the clinical therapeutics in the near future. So this epigenetic deprogramming will be expanded a lot and clinically available in pretty soon. That's my expectation. This is end of my presentation, and thank you very much for listening.